Hello, everybody. Welcome to another session of the Bioinformatics and Genomics from data analysis to AI applications. Today, we're going to cover a little bit of a niche topic that may be something that you all may be interested in, and that is de-novo detection and annotations of transposable elements. You may be a little tired of hearing me talk, so here today I have brought with me a colleague of mine, Clément Goubert. I have butchered the name like I often do.
Good enough. Michele.
And Clément has been a colleague of mine for a while now. We have worked together on a few workshops, a couple also including the last fall semester webinar series as well. And I have invited Clément to come back simply because his knowledge on transposable elements is extremely valuable. And what we have for you today is a little bit of a showcase on transposable elements and detecting them. We will be giving you virtual machines that you may be able to work together with. But before I get ahead of myself, I'm going to turn the mic over to Clément. And by all means, Clément, the floor is yours.
Okay, thank you, Michele. Actually, would you like to do a demo on how connecting to the machine before we start that? So it's set, and then I do my introduction, and we can start with the first part.
That works.
All right, let's do that.
Muted. Okay. Let me share this with all of you online, at least. For the ones that are in person, please do connect to the Zoom if you want. But otherwise, this is the document that we are going to be using, and I'm going to show you a little bit of how this works. So if you've never used the SSH before, SSH stands for, excuse me a second, Carlos, can you go and fix the computer? It reboot in 2 minutes. First class, yes. Thank you. If this is your first time, apologies for that. If this is your first time hearing of SSH, SSH stands for Secure Shell and allows you to connect to a different computer using the command line interface. So now I'm going to show you a little bit of how you can connect to your computer. I want to state that these computers do not have a GUI and therefore are only command line. So you will not be doing any tunneling or whatsoever. If you have seen earlier, actually I should share my entire screen. Here we go. The way that you log in is by doing SSH user at IP address. Please do put here your actual, not user, but let's call it real person. And that's how I've always called these sort of columns. And here I will put in my name. And so no one else can join in, but
Too late.
too late.
To be user one?
No, you can be instructor. It can be two people in the same machine. Don't worry. So what you will do is you will go to the user, copy your username and go back to the terminal and do SSH, put in your user at, copy the IP address. And as you can see, the IP address follows the at. So you're going to paste here your IP address. And then you're going to paste in your password, where you're going to copy the password over here and paste it within the shell. Press enter. And if you get a message similar to this, let's make it a little larger, then that means that you are within the machine. The machine does come with the tools necessary for all of us to follow Clément and his workshop for the day. All of the tools and data will be under data. So you know what? I'm going to let you drive. I think you have it from here. So I'm going to stop speaking. I'm going to stop sharing.
Yeah, and I will guide you again through the connection again, and I can see, you can see myself a million of times now. It's amazing. All right, so as Michele was introducing me, I'm someone who works a lot with transposable elements. First, I come from a evolutionary biology field, but in the last 5-6 years, I've been focusing on bioinformatic methods, and in particular, methods that have to do with analyzing transposable elements. So the workshop today is going to be in 3 main parts. It's going to be a short introduction about transposable elements, what we should expect to see when we analyze a new genome. And then we are going to break down the analysis in 2 parts. The first part is what we call a de-novo detection, and I will explain you what it means. And the second step is going to be the annotation. So once we have a collection of repeats that we think exist in the genome, we can try to find where they are in the genome. And this is the second step. So let's get started. Oh, I did already I should share my screen. Perhaps that would be even more, more informative for everyone here. Okay, I can see that we can see the slides, so that's great. Okay, so and if you have any questions, please drop them in the chat. I think if I don't see them, Michele will won't miss to tell me.
You can also look behind you in the back.
Hello. All right. So first, what are transposable elements? Just a basic introduction without going into too many details. A transposable element is a repeated DNA sequence, which initially has the ability to replicate itself and reinsert itself somewhere else in the genome. So they are mobile, repetitive sequences, and they are almost found in any eukaryotic genome. So if you have to analyze a eukaryotic genome one way or the other, it's very likely that you're going to at least stumble upon one transposable element, or you're going to have to deal with them. So I think I always like to start with this because the goal here is what can we expect when we have a newly assembled genome, and in terms of transposable elements, how many are going to find, where are they, what are they? And so I like to talk about this T life cycle. Initially, we have an active T, so a T that is able to produce the enzymatic machinery for its own replication. That's going to insert in the genome and colonize different loci in the genome. So this is what's happening here. But this is, you can imagine, if it's unchecked, it could be very detrimental for the host. It could disrupt genes. It could disrupt regulatory gene networks. And often T insertion might be deleterious, and if they are deleterious, then they aren't going to be passed on. So the genome usually evolve, I should say, defense mechanisms that are mainly epigenetic. And these defense mechanisms lead to a previously active T copy to be inactivated, first transcriptionally, but then eventually they start to accumulate mutation, point mutation, insertion or deletions, and it's going to make this copy unable to produce the enzymes that are necessary for the jump. So what we are likely to observe in the genome is a landscape where we have dispersed repetitive DNA with some degree of variation or degradation compared to the original insert. So we will have different T that have different level of degradation. So some are still active, some are fossilized, as we say. And there are several mechanisms that we won't discuss today, but for example, aging, stress, disease, hybrids, things like that, that can lead to reactivation of T. So we talk about wave of amplification of T through the evolution of organisms. So this is mainly for the theoretical biological context. But for the task that we are interested in today, which is finding T in a newly assembled genome, what are the important steps that we need to go through? So the goal is to identify repeated sequences in a given genome. And for a given type or for a given sequence, what we can identify is copies that belong to what we call a family, or at a smaller resolution, maybe a subfamily. So this is a group of homologous sequences that we can basically find homology between them and align them. So they can have some mutation, but they are globally alignable. And there are really 2 ways to create families from copies that are dispersed in the genome. One way that would be the ideal way, but it's often difficult to completely perform, is using a phylogenetical approach. So we make an alignment and then we create a phylogenetic tree. And we can decide that, okay, this node is the node where all the members are similar enough, and this is my family. But practically, the field has been developing some heuristics to try to automatize this the discovery of family. And there is a very famous rule in the T world that's called 80-80-80, because it says that we can group two sequences into a single family if their divergence is no less than 80%. Sorry, their identity is no less than 80% or more. Sorry, so 80% identical or more at the nucleotidic level. And then that the one sequence, at least 80 base pair of one sequence align in the other, and that they align within 80% of each other. So there is like all these parameters, these three parameters that are described here, where basically it says, well, they need to be looking alike, right? And this rule has no real biological bearing. And so it's really a choice that is made. Well, everybody does that, but it doesn't mean it's optimal. So once we have created some sort of grouping, clustering, or phylogenetic grouping, whatever, created some family or subfamily, one way that is the traditional way to represent this family is to create a consensus sequence. That is the average sequence. So if there are bases that are variable among the copies, like here, we would simply pick the most abundant in the multiple sequence alignment. So it's a majority rule that is used to build a consensus. So you will hear a lot of people working with T talking about consensus sequence, and this means it's like a representative average sequence of a given family. More recently, and with the development of methods that are based on what we call HMM profile for hidden Markov model profile, just you don't have to remember all that. The idea is that instead of losing information by, for example, saying, oh, okay, we're going to keep an A here, we can say, oh, A occurs at in 2/3 of the case, and C occurs in 1/3 of the case. So this is another way to store a representative, maybe in a probabilistic sequence. It's called an HMM profile. And this is a representation here, for example, with a logo where you have all the different possible bases in the alignment and their probability. And when you use one or the other, the second one is usually more sensitive. That allows you to find more degraded copies, or it's at the cost of a higher computational cost. And it also makes a big difference between existing database of T. We have a legacy database called RepBase that unfortunately has been paywalled for a few years now, but traditionally has been storing consensus sequences. And DFAM, which I very in a limited amount work with when I came, we store HMM profiles, but from an HMM profile, you can infer a consensus. So it's just a way to store more information for a given family and be more representative. So this is about what are T families and what's the concept about finding them. And so this basically represents the first step that we are going to do, discovering, like finding families in the genome. But then once we have found families and we say, oh, this is the representative sequence, or this is the HMM model for this family, we might want to say, okay, where are the copies of this family in the genome? So we might eventually get a file, for example, that gives us for each family their coordinates of the coordinates of different copies in the genome. So usually for that, we use what we call a T library. So this is a database of either consensi or HMM profile, and with different tools, we can search them in the genome, and we can identify copy and divergence between the model and the copies, which help us, and we'll see later, to do some biological inference. So the goal eventually is to identify all the copies of all the families that we have been able to identify. So this is the annotation. So here I've summarized a strategy for discovering and annotating T in a genome. Just a little preamble. Usually it starts with thinking about your assembly strategy, because transposable element being very repetitive by definition, but also having somewhat a size, quantifiable size, maybe 300 base pair at minimum, up to 10 or 20 kb in length. If you sequence with short reads, for example, you will miss probably a lot of transposable elements. So thinking about when you think about transposable element, it's good to know and think about your sequencing strategy. What we're going to talk more about today are the points 2 and 3. So yes, okay.
I have a question.
Yeah.
So
Yes, doing short reads, you might lose a lot of the transposable elements, but doesn't that increase the accuracy?
Oh, well, in terms of sequence of
Yeah,
for a given yes, you will. The traditionally, right, short reads are more accurate. The problem is you are going at the T level. What's probably the most important is to identify the boundaries of an insertion, so the beginning and the end, rather than the exact sequence. You can always refine that, and it's a good point. You might, for example, especially with the first iteration of noisy long reads, like the first version of nanopore, you might look at the alignment of a family and say, oh, they look very different from each other. Maybe it's a very old family.
Yeah.
And yeah, this is something to keep in mind, indeed.
It's quite trendy now to do long reads
Yeah.
first and then shorter reads to increase accuracy. Okay.
Absolutely. But overall, in the T field, it's been a blessing to have this long read data set, because we really can, we see that, for example, the estimate of T content in percentage of a genome, right, were a little bit or quite dramatically underestimated with short read. So this is usually an advantage. So yes, what I told you before is that we're going to break down things into the De-novo or ab initio reconstruction of T family, and we will look at an example using probably the most used tool. It's called RepeatModeler2. In very little, just very little participated in this. Rather, I should say a student of mine developed, contributed to the development. So it's the algorithm I'm the most familiar, but just know there are other things out there. And then for annotation, there is mainly one tool, that is RepeatMasker, that have been there for, I think, since 1996. And that is the most widely used tool to annotate T. So we'll look how to use that. If you read a paper with T annotation, it's very likely that RepeatModeler2 and RepeatMasker had been used. And then if we have time, we will look into a few downstream analyses, what we can do with the results of RepeatMasker and RepeatModeler2. So if there is no question at this point, we are going to start looking at RepeatModeler2 and what basically is the ab initio reconstruction of T families. So when we have a genome assembly and we are like, okay, what are the T in there? What are the T family that exist? You have different strategy. You can skip this de-novo search and directly try to look if there are T that are known from databases. So it's the most basic approach. This is maybe precise, but you're going to have a very low recall, because probably many T in your new genome assembly have never been found. So this is recommended if you resequence something, but maybe not if it's a new species. We have tools that are specific to different kind of Ts, in that they are able to recognize several structural patterns or specific enzyme that specific T bear. So this is good if you are very interested in one type of T. For example, I show here LTRs that are type of T. But once again, it's not going to be comprehensive because it's very focused. And then the last category is completely agnostic upon what they're going to find. And the only goal of this ab initio method is to find whatever is repeated, and perhaps try to improve on the refining of this family that we are discovering. And so individually, as I say, it could be limited, but usually in combination, this is the best strategy. And because I don't have the time to go into the details and it's more hands-on workshop here, I really recommend to check out this publication, where one of a colleague of mine, Jessica, went through, and she used to work at DFAM, this database. Jessica go through all the different pipeline that existed at the time. There's a little bit more now, but it gives you a really good idea of how these different methods are combined to reach our goal, that is identifying repeated families. And so this is the content of the paper. For time, I'm not going to go through this. And so with that, I think we are going to now start put our hands on the data, and we are going to do a reconstruction, a discovery of T families using RepeatModeler2. So for that, there is a link that I think I sent you, Michele. I don't know if we have shared it with the group today, where you will find in the link, you will find these slides, the handout that is simply the list of command line that we are going to use today, as well as the one data set that we're going to use. So what I'm going to do now is switch to the handout. That is here. Perfect. Okay.
The link, there is.
There are 3 files, right? One is the slides, one is this handout, and the last file is a chromosome of the Saguaro genome. Okay, so if this is good for everyone, there's going to be a quick intro for that, and then we're going to already start computing. And then as RepeatModeler will be running, we will discuss quickly about what it's actually doing. And we'll do the same with the second step. So I thought that it would be fun to do this analysis on the Saguaro genome. And as the Saguaro genome is also gigantic. So what we are going to do today for time is just pick a small chromosome, or actually a contig of the assembly, because this genome is very complicated to assemble. And we're going to take a 2.1 megabase contig and use it as a toy example, thinking, oh, this is our assembly, this is our genome. So this is the pipeline. I've discussed it already. We have our genome in FASTA, and we're going to give it to RepeatModeler2. That's going to find our repeats. And then with these repeats, this what we call a repeat library, we give that to RepeatMasker, who is going to go back to the genome and annotate the repeats. So RepeatModeler, I talked about it a little bit already. You have a link here if you want to know more or learn how to install it and download it. But for us, it's going to be very easy. So what I'm going to try to do, if that's not too small, is to have both my terminal and the handout. Yeah, that seems good. Maybe I can increase this a little bit. If the command line I'm typing ends up being too small, just let me know.
Looks good.
Yeah. Okay, so the first step is what Michele was telling us earlier, is to connect to the virtual machine. I have just done that before, and so I'm already in. But if you need 1 or 2 seconds to do that, I'm going to wait maybe 15 seconds.
Okay, 15 seconds. Yeah.
Just making sure. You can also put a little thumb up in the chat if you are if you have managed to, if you are connected to the virtual machine. George is asking, how do we know which IP to use? You just pick the one you want. And you put your name. If there's no name, it means that it's available.
Exactly. Thumb up.
Thumb up? Oh, okay. I guess that's all the thumb up we were waiting for. Okay, so the first thing to do is we're going to create a directory where we're going to store our data. It's very simple. We type here, data, boom. It already exists because I did it before. But it shouldn't. This shouldn't happen for you. If I do ls, I see that indeed I have data. And so once you have created data, we are just going to launch a Docker container. The Docker container allows us to run many programs without having to install them. And if you just copy this command, it's going to open an interactive session with the Docker container that contain RepeatModeler and all the dependency that we have. And we'll at the same time indicate that the data directory in our machine has to be placed somewhere precisely in this Docker container. Don't think too much about that. We've set up so it's very smooth for you. But if you have particular question, don't hesitate to ask, of course. But basically, now that I have entered an interactive session on my Docker container, if I do ls/data, I'm going to see, normally you shouldn't have anything, but this is going to be the directory where we're going to download the things. So what we do is cd data. Oh, sorry, /data. Oh, I should learn how to type. Okay. And here we are going to download from the link I sent you, the one chromosome from the origin. So I'm just going to re-download it. It's okay. I'm sure there is tons of room on this server. Not really.
Yes, there we go.
Okay, so we can do ls to make sure we have this file. You should only have this file at this point. And we can look at it. For example, if you do head of the file, oh, it's a FASTA file. That makes sense because it's a genome. And just by eye, you can see that here we have some sort of tandem repeat here. But this is not a transposable element, it's just some tandem repeats. By the way, actually, RepeatModeler and RepeatMasker are able to identify also this short repeat or tandem repeats, things that aren't transposable elements, but that are repetitive DNA. Okay, so RepeatModeler has two steps. One is equivalent to indexing the genome. It's called build database. And the second is just basically running the program. So when we do build database, we have to provide a name, so our row here, and our genome in FASTA. This should be very quick because it's not a very big sequence. But when it finishes, it just tells you how many base pair, which is here the length of this config, have been added to the database.
The database is a question. Is a where is it?
It's an executable. Yeah, it's an executable from RepeatModeler. So in this Docker container, we have RepeatModeler in the environment. So it's installed such as when we type RepeatModeler or build database, it's showing up. Perhaps when you install it, but I really recommend to install it using this Docker because it's really smooth and easy. If you were to do a manual installation, you might have to find where, in which directory you've actually installed it, right? But in the RepeatModeler directory, you will have RepeatModeler and build database, as well as other tools that we will be using today. So now that we have built our database, we just have to run the program. And while it's running, I will show you what it's actually doing. So you just copy and launch it. It should take approximately 10 minutes, which is the time I need to show you the rest. So good so far? Make sense?
Yeah.
Okay. It's running. Thanks to you, Mikael.
Thank you.
All right. So what is RepeatModeler2 basically? What does it do? So RepeatModeler is a tool that is going to, in big summary, sample your genome, identify, compare these samples to find repeated things, and refine each family of repeat into a representative family and store it into a database. So there are different, those sampling and compare and identification of family is made iteratively. So there is a first round that is done where some pieces of the genome are sampled. And as I represent here, we have the genome in black and two different T families. The copies have different sizes because, as I was telling you before, as the genome evolves, mutation accumulates, deletion, and so we have this sort of remnant of families. So we do a sampling, and then we are going to compare. We're going to compare the sample sequence to each other to identify repeats. So in that case, for example, we had enough repetition of the blue family in our sample to align it and identify a new family. Okay, so from this sampling, we align, comparison, and then we infer a model, which could be either a consensus, right, or a HMM model. Here, it's just stored as a multiple sequence alignment, but it's not important to memorize that to successfully run the program. However, you would say, well, sure, but I've only found one family here. But don't worry, I'll say, because we put this model in our T, what we call the T library, and we keep going. We go back, take another sample. Oh, sorry, this is here. We go back, we take another sample, and maybe in this second sample, we have more sequences from another family. And of course, at each round, it's not one family that we're going to find. It's like hundreds, maybe sometimes thousands. So then we have the green family that seems to be more represented, and also we would like to avoid to reassemble the family, the blue family. So what's done is that we are going to, by homology with our library, try to identify previous instances of this blue family here. So we're going to mask them. We're going to replace the base by ends. And then are only left here repeated the green sequences. And so similarly, they're compared to each other, and we can infer a second model, et cetera, right? Where this loop basically is occurring up to 6 times with RepeatModeler in order to kind of make sure that we have sampled enough of the genome. It could stop before if the algorithm, maybe let's say after 3 iterations, there's no gain in new families. It can identify that that's it, we found it all. Mm-hmm. The bigger the genome, the less true this is, mm-hmm. And also it's a bit paradoxal, but the less repeated the family, the more run you're going to have to do because you're going to really have to mine your genome for repetition. So if you have a lot of repeat, maybe in a few runs you have already identified most of it. But if you really have maybe 10% of repeat in the genome, then you're going to have to mine this genome even more to identify them.
What I know this is not your first rodeo, so what organisms have you tried this on?
Yeah.
Apart from the saguaro?
So apart from the saguaro, I've done that in mosquitoes.
Mosquitoes?
I've done that in Curculionidae, so these weevils, the little insects. I've been doing that in plants, so like roses, and I've done it so many times. I don't remember. But no, and I've seen people doing it, you know, in fish a lot, like a lot of people do it in fish. It's designed to work on any organism because it really don't have a preconception about what type of element it should find. So as long as it's repeated, it's gonna take it and kind of look at it in more detail. So here, the last slide, I'm just gonna take a look quickly at our calculation. Where are we? We are in the round 2. Okay, so I think after this, we're gonna hit the step that is going to try to, oh yes, exactly, refining, and then we're gonna let me talk about that here. So I talked about this. What I showed before was really in a really high level, but in more details. What we can see is that the first round is performed by the 40 megabase pair sample of the genome. So it's a very small sample. And it's going to identify with TRF or ultra, it's going to identify these non-T repeats, so a very short tandem repeats, so low complexity sequences, and put them in a bucket. But then with what's left, there is a software called Repeat Scout that basically is a fast k-mer comparator to identify really what is very similar and very repeated. So this first round is basically to identify what's easy to find. And then as we keep going, the round 2 to 6 are made with a larger sample size. And after masking of simple repeats, we have another algorithm that's called recon that is much more sensitive. It's going to take much more time to run, but that is this recon software is able to identify much more diverged sequences and perform all VO comparison and clustering to identify families. So as everything goes through, we we add families to our bucket in our T library. RepeatModeler2 differ from RepeatModeler1 by the addition of this LTR structural detection. LTR are long tandem repeats element that often can be harder to identify with this traditional method. So we just added this LTR specific tool that is really performant at finding LTR. However, this is occurring after all these rounds, and it sometimes finds things that have been found with the base algorithm. So there is a merging and redundancy removal step that is performed in order to end up, the goal of a T library is that there is no two sequence identical in this library, because it's our goal to actually put them in buckets that are non-redundant. And as you see here, the last step is the classification. So often classification of transposable element is confused with annotation. Classification is just, here is the sequence of a transposable element, tell me what it is. And so it could be, oh, this is a line element, this is an LTR element, class 2 element, etc. So this is a task that is actually very complicated to automate. Usually it's done by manual curation. But sometimes you can have hundreds or thousands of families. So in this software, the classification is based using a high-quality database of protein and nucleotidic sequences. So it's going to help you identify known families, or at least look-alike families. But of course, if you have something completely different that have never been found, it will simply be labeled as unknown. But you have the ability to go further and then do this manual refinement later. Okay, so let's go back here. We see that we are actually at the repeat classifier step. So we are at the last step of the pipeline. If I go up in the log, we can just see what it's been going through. The first round. And the first round, you see, collecting 40 megabase per, I didn't lie. Running repeat scout. Then we do round 2 with a larger sample. And it's going to run. And then we have recon that should be called somewhere here. Up. So you can see it's a call to recon. So this is this more sensitive algorithm. And then once this is done, you see that here after 2 rounds, we stopped. It didn't go 3, 4, 5, 6. It has identified everything it could at least. And then you have the LTR structural analysis that is performed with the tool called LTR Harvest. And finally, we are classifying. So this is taking a little bit because this is just homology, but this should finish like in a 1 or 2 minutes. So that's a good time if you have question, because we are going to continue right after. For next time, I will try to have my speech that ends exactly when it ends, but I need more practice, Michele.
Third time I have you do this.
The second time, Michele, it's just the second
But
time.
I've seen you do
Yeah.
this one elsewhere.
Yes.
And I'm like, you always run into, okay, now we gotta wait.
No, I mean, I don't know. It's you said, that's it. That's done. All right.
40 seconds.
So before keep going further, maybe we can look at what the program is giving us. So I have way too many things here. I should have removed everything. But okay, there are two files that are extremely important. One is called sarofamilies.fa and one is sarofamilies.stk. Both are the same thing, just two formats. FA is going to be consensus sequence derived from multiple sequence alignment that here we have in the .stk. STK stands for Stockholm, and it's multiple sequence alignment that is used at DFAM, but it was also used at the time of PFAM, for example, and RFAM for RNA. It's just a way to represent multiple sequence alignment. And it's used by, if you know the software HMER, that's used Stockholm file to create models. So if we look, for example, at what we have, so for example, we can do more of sarofamily.fa. We have a FASTA file with different sequences, and each the most information we can get from it is in the header. So for example, this one is called round 1 family 4, so it means it's the 4th family found in the first round, and well, it's unknown. And that makes sense because the Sawaro being an organism that don't have a lot of similar organism in the T database, we don't expect to find a lot of T being classified here. Okay, so this is one consensus, this is another one. And so if you're interested to see actually which sequence contributes to a given consensus, you can look into the STK file. So if I do that, I'm gonna find much more text-rich type of file, but eventually what you see is that we have here all the different copies with their location in the genome that have contributed to creating this consensus sequence or this multiple sequence alignment. So each family is represented both in the basically, if you name remember when we build the bit database, we called it Sawaro. So it's going to be the name you provide, families.fa or family.stk. Okay. For time, I think I need to move on for annotation. What's very important here is that we are going to reuse this family.fa here as an input. So the FASTA file is going to be used as our database for the next analysis, that is masking the genome or annotating the genome with RepeatMasker. Now that we know the set of family that exists, we can try to find them in the genome. So we can do a quick thing that is, for example, counting the number of family that we have identified. So if I do grep -c of this little superior sign, it's going to count every header, so every FASTA sequence or every family. So we have 20 family that we have created here in this with this analysis. This is probably way under the total number of family that we should find in the Sawaro, because this was just a very tiny chromosome, right? So just keep that in mind. Normally, and especially in plants, the number must be much larger. Okay, so what we are going to do now is using RepeatMasker to use our T library, our models, to find them in the genome. What is very important and what distinguishes RepeatMasker from other tools, for example, BLAST, you could say, well, why don't I use BLAST or why don't I use any other aligner? Actually, by default, RepeatMasker use a slightly modified version of RMBLAST10 that is just a tiny bit more sensitive. But the very important thing is that it does a competitive search between all the families that you have identified at the same time, because you could have two families that are similar enough to generate alignment to the same locus. However, there might be one that is actually the real family that has a copy versus another one that is just a cousin family, let's say, right? So this is very important because some T, and you're gonna have domain within T that are very conserved, maybe enzymes, versus non-coding parts that are more diverged. And different family can have a lot of similarity between subregions, for example. So it's very important that when you want to find T in the genome, you use a tool that does this type of competitive search, and RepeatMasker does that. In the simplest form, RepeatMasker, you give it a genome in FASTA and a library of T, either in FASTA, as we're gonna do, or in STK, oh, sorry, not STK, but HMM as HMM profile if you use, for example, DFAM. And what you're gonna get as an input is a genome that is masked wherever there was a T, now we have X's or N's that are gonna be X's usually. But not only this, we will see that later, but we're gonna have ability to have soft masking, so which is we put in small letter in lowercase region where we have T's. We're gonna have tables with the location of all these copies, so we'll see that at the end. I will show you how it works, but before, let's get it right. So for this, let's just retrieve our handout. There, it is here. Okay.
Let me scroll all the way back.
Okay. This is how it goes. All right, so we've done this, we've done this. All right, I did all that without looking at the handout. Okay, so now we're gonna run RepeatMasker. So the command RepeatMasker calls the program and a bunch of options here, I'm gonna detail in a minute. But what's crucial here is that you indicate lib, sour family, .fa. So this is really our new input. We have created this file with the RepeatModeler, the program before. And so now we are going to use it as an input. One of the input. The second input being our genome again, because we want to annotate it. This option is like, we're gonna use one CPU. And this is way enough to do that. And while it runs, I'll show you the other options. So the S option is recommended. It's called a slow search. It's it's basically it's the most sensitive search with RepeatMasker, and it's what people that are using the program recommend to use all the time. -A, it's going to export alignment in the format that's going to allow us to create some downstream analysis. So in some cases, if you, for example, storage is important, which this is not dramatically huge, I always recommend to use -A because it allows you to do a little bit more downstream analysis that you could if not. PA is the number of processors, right? We talked about that. And you have here, I put a link to the documentation of RepeatMasker. You can also find all the options by typing RepeatMasker-H. And in any case, if you have a problem with RepeatMasker, I will copy that in the chat, if I find it there. You can send an email to help@dfam.org, and I will receive it as well as a colleague, and we will be happy to help you. Oh, it has finished. Perfect. What a perfectly tailored session, right? Look at that. So the way to know that RepeatMasker has concluded perfectly, right, is that it says, I go through all these processing output cycles, and then it says done. That's very explicit. So now you know it has been working. So let's look at the outputs. And I've also in the handouts I have put the explanation here. It's a little bit small, but I'm gonna go with you. So you can find here that you have your genome, that was our input. And we have here our library of T of consensus, that was our second input. Now let's ignore this, but we have this dot align. So that's one file that contains all the alignments, as I was saying, talking before. It's important for downstream analysis. This is sort of archive that is necessary for debugging. So if you have a problem, often we ask for this file. The masked is the same as the genome, but with all the T. So let's look. You see that we have introduced, well, these ends are not, yeah. You see that we have all these ends that appear in our sequence compared to the actual one. And what it means is that wherever there is an N, there has been T's. So sometimes you have an option in RepeatMasker. You can say annotate with N's or with X's if you wanted one or the others. Here it's N by default. Okay, so this is an important input. But so for example, you could use this input if you want to search for genes. So when you do the de-novo gene prediction, you want to mask the T before in order to not identify gene as actually T that where you say, oh, this is a gene, but this is actually a T. So often it's recommended to mask T and then you do the gene prediction. So this is an important file, but perhaps the most important is this .out. The .out is a table that indicates where in the genome are the different copies. So we can look at that a bit. Okay, let's keep going. So basically, and you can find on the website I have linked here, you can find all the explanation about the different columns here. But what it says is that for every position in our config, so this is our config, this is the coordinate in the config, we have this family, round 1, family 7, that is unknown. But it's not very exciting, as I was telling you. There's not a lot of known family in this genome. But we can keep going. Oh, here is a line 1 element, for example. So here we know that in this config, from this position to this position, we have in the complementary strand this family that is a line 1 element. And then these are the coordinates relative to the model, the consensus sequence. I don't want to go into too many details here just for the sake of time, but if you have problem interpreting these outputs, please reach out to me. I'm very happy to do that. This output is the native output. There is a convert version in GFF format that exists, so we can look at it. So this is a more conventional type of file of output, which just says the same thing, right? This is the config, this is the position in the config, this is the divergence and the strand, and this is like the details about what type of family is that. So this is sometimes usually what people use if you have your genome loaded in a genome browser, and then you want to overlay the track with the T. This is what you would use. And finally, the first thing that I like to look at when I finish a run with RepeatModeler is the dot table. It's a sort of small summary that gives us some indication. So for example, it says, okay, our input was this file, it was this length, and we have a GC content of 36. In this config, we identified that many base pair being repeated, which is 20%. So if this was a complete genome, one could say, well, it seems that the T content of this genome is 20%. So that's probably the most basic type of result. However, this is very like a high-level overview. If you want something a bit more detailed, there are a couple of commands that we can run, native that are like with tools that are available with RepeatMasker to simply get some summary statistics and some more detailed output. So we're going to use this. There's a little tool that's called build summary. It takes as input this main output, the dot out that I was talking about, and it's going to create a summary. That's very quick. And if we look what's in our summary, we have some table, but then eventually we have this table. Well, this is the simple repeat, so there's going to be a lot of them. There you go. Here, what's the most interesting? So these are our different families, and these are going to be the number of occurrences in our genome, and these are going to be the number of base pair that total base pair it represents, and then the associated percentage. So you see here, this 20% is the same that we had in the summary table, so everything checks out. And then, whoops, sorry. Right, so this is here, we have only one sequence in our genome. So that's just, but if you had many chromosomes, for example, you would have the summary per chromosome. So this file is important because we can use it a little bit later. And probably if you want to, when you publish, and then if you want to show, for example, oh, okay, we have that many of that family, that many of that family, this would be this type of file that you could analyze and make your graph out of. Next, yeah, we have 10 minutes. I think it's enough to do a very cool analysis. And I think for that, I want to show you a little bit what we have here. So let's skip the details about RepeatMasker. But if you have questions, feel free to ask me. There's also a more in-depth tutorial that you can access via this QR code. So if you have the slide, you have the link. That's Jessica, who worked with the people at RepeatMasker for a while, that she can explain you a lot of details and little insider tricks. There's, so we discussed about the output, we did it. So I have compiled everything in the slides. Sorry, I'm going maybe a bit quick, but once again, feel free to reach out to me for details. So I was telling you the GFF can be loaded in a genome browser. You can see your T relative to the gene, for example. Okay, we've been looking at all that. Perfect. So downstream analysis. We've talked about that. I wanted to tell you about this thing that we call T landscape. So what's very interesting when you analyze T is not only knowing what's the percentage of T in the genome and the breakdown into different type of T, but also what is the relative age of these different family. When did they enter the genome? When did they expand compared to each other? Because this thing is dynamic. It occurs as the species evolving. And so at a given point in time, you have only a subset of T that are active in the genome before they get silenced, and then another maybe family will come later. So ideally, with our alignment, well, what we've done with RepeatMasker, we get copies that show some degree of divergence between their average sequence, the consensus that we're talking about. So very old family will have copy that have spent a lot of time in the genome and so accumulated many more mutations than a younger family. So if we look at the average divergence between our copies and the consensus, an old family will have a higher number of copy with high divergence, while a younger family introduced more recently or that has expanded more recently, will have copies that are more similar to each other, so more similar to their consensus sequence. And so by doing this analysis that's called T landscape, we are going to plot the distribution of the divergence for each family according to the person. We're going to have the distribution in percent of genome according to the divergence. And the way we interpret this is that as we go far to the right here, we are going back in the past. And so we see these waves of repeats, right, that happen. And then because we have done a stage of classification, we can even say how these repeats occur there and there. So for example, what we can see here is that the blue type of repeat, line 1, seems to have been occurring or at least expanding at a time that is before the red one, that are this DNA CMC type of family. And perhaps those green ones that are LTR type of elements have expanded more recently, but not as recently as the DNA. And so that's showed the different wave of amplification. Of course, you can note here that we have still some unknowns, and this is just because this is a genome that has been just not so much explored yet, and we don't know about all this T. So if you are bored, you can just download this data and try to manually identify what they are. So in the few minutes that we have left, let me show you how to do that. Not that. Okay. So we are here. We just need to perform these 2 steps. What's so funny, Michael?
You're rushing as the clock.
Yes, I'm rushing as the guy. I think I can do it. I can even wait 1 minute like that. No, but I'm not going to do it. So, so the first step is to calculate this divergence from the alignment. So we have this file I was telling you, it's always important to do it, the -A alignment, because from there we can calculate this value of divergence. And we're going to store it in this file. Oh, we're going to say, hey, put it there in this div sum from this file, the .align. We do this. This is very quick. For real genome, it can take several minutes, even, maybe an hour or 2 if this is huge. But for this is fine. We can have a quick look at what file we've created. It tells us, basically gives us for every type, every family we have identified, what is their divergence, the average divergence. Okay, so this is good. And let's keep going. So now we have the create repeat landscape that take as an input the file we created before, so the divergence, the distribution, because we're going to draw the distribution. And we can give a title to it. And what's important is to tell it what's the size of our genome, because then it's going to give us the result as a percentage of the genome. And the output is an HTML page with a graph, and we're going to look at that. So run this. It's already done. Very quick. And if someone was wondering, why do I have a second terminal open just right here? It's because this terminal window it's a new terminal window that's connected to my computer. And so I'm going to pull oh my God, I didn't save the, see, yes. We're going to pull the data from our virtual machine to our computer so we can look at it. So to do that, it's the similar process of what we've done to connect. Instead of SSH, we're going to use SCP for SSH copy. You're going to have your username here, the IP of the machine, and then you have to add this here, which basically says, oh, I want to grab this file that is here in the virtual machine and put it here in my computer. So we do this. It's going to ask our password. So I'm going to go. I didn't copy it, of course. Up. Go. Now I've downloaded the file, and I can just open it. There it is. So what do we have here? Actually, you've seen this file before, but now we can hover over it and see different things. Let's start with the more boring part here, this pie chart. This pie chart is simply the breakdown for the genome level of each larger family, what we call sometimes order. So we can see that we have nearly 80% of our input sequence that is unmasked. So it means that it's non-repetitive, or at least we haven't found enough evidence for copy at the first stage to create families, right? And then we can see the breakdown. So we have 2.8% of simple repeats, 1.9% of line L1 element, LTR that are very abundant in plant usually. We have 6.8% here, and it's the most abundant class here. So that checks out. Some DNA element and some unknown element. So this is very common, and you shouldn't be sad of having unknown elements. It means that you are dealing with a species that is new enough, and that's probably potential nice discovery. And then on the left, we have this landscape that we were talking about. It's not working for you, Michele?
Yes, but it's not your problem. It's from my side.
Yeah, but anyway, you should have this, right? And so this, as I was telling you, basically, if I were to do a very simple interpretation, I could see that we have overall a recent amplification, or at least relatively recent, of amplification of TE. If you have a molecular clock for a given species or a given group, you can actually use the molecular clock to try to find in million years. But there's a big assumption here. It assumes that all the TE will evolve at the same rate, and this is not really the case. So you could do it. It just interpret with a grain of salt because it's probably quite limited. It gives an idea. My best advice to date T insertion is to actually, if you have the ability to compare different species and know when the common ancestor of the species are, then you can identify presence and absence of orthologous elements. So it's really the way to know, okay, well, until this time in the tree, there was not this element. So I know that it's older or more recent than this time. This is for another advanced workshop, I think.
And it's important, if I can, we're out of time, so maybe I can probably talk about this later.
You wish? We have 1 minute, 30 seconds.
Do you have anything else?
I have nothing else to show you guys. That's it.
I wonder if there is also a stuff to do with talking about the rate itself, you know, thinking that perhaps you'll have to look at reasons, environmental reasons, for example, that will push for a specific let's call it evolution. So yeah, I wonder if there is any research out there that is looking at this and it's also looking at the evolutionary rates related to environmental factors.
Yeah,
well.
Actually, a big part of molecular ecology is kind of working on these things, like not only with T, right, but other type of mutation. Yeah. And T have been shown to provide adaptation, but often this is at a single copy in one specific context provides something. So it's usually at the copy level that you're going to see interesting stuff. But there are some examples out there where it's been shown that indeed an expansion in TE as fast as is correlated, right, with radiation and adaptation.
Right.
It's a tricky field. There are some there are a bunch of research done on it,
Yeah.
but the results are not always the same, right? Depending on one model to the other. I think early on the expectation was, oh, it looks like a very exciting evolutionary system where, yeah, maybe TE are, whenever there's a sort of strong ecological constraint,
Yeah.
you make them explode. But it's also very risky because you take the risk of breaking the genome. So it's not that easy. It's in some contexts it can occur, in some it don't. And so, yeah, it's this type of thing where you can do with this type of data if you have environmental data associated.
Yeah.
It's very interesting to look at it because it's still a very active field in research. So there you have it. I think
Was getting the same error
issue.
as I was.
Okay.
So I wonder what I think we cannot just do SCP.
From
maybe we need to change a little bit the.
I mean, I just did it in front of you guys.
I know. Maybe you did it and it worked.
Yeah.
Maybe it's not tilde slash. Or it's just slash.
It could be just slash. It could be. Yes, I'm sorry about that. It can be actually, because what we've done, I think we might have mounted to slash.
No, it's not.
It's not? Okay.
Well, we'll figure it out next
Well,
time.
George, I know you. I know where you are in the lab, so I can show you. It is threatening. I am threatening. I know where George lives. Yes, of course, Michele. Sorry.
All right. Thank you very much, everybody, for being here today. And I do understand that just in case you may want to try this again later on, you can find all of this information shared today at the, I'm going to share my screen real quick. Ay, ay, ay. My slides are getting in the way. There we go. Oh, that was too late. You can find all of these slides that we saw today at Exploring Tools for Data Analysis and AI Applications. This is where we have all of our content. If you were to just scroll down and click on the material, it's going to get you to this little page which Clement was showing earlier. With that, once again, thank you very much for your attention, and I hope to see you next week for our last session. Thank you all for being here today. Bye.